Thank you very much. Presenting on behalf of colleagues, there are fundamental problems with YAM research, where we really wanted to investigate underlying QTNs and candidate genes for economic traits of interest. So that actually motivated us to undertake this particular study. This is the order of my presentation. So YAM obviously provides income and food for over 300 people around the globe. Is very important. Development of clones, is really important because it contributes to food security and it has other applications. However, the breeding efforts have less explored the genetic basis of tuber yield and virus resistance traits that are quite needed even to fast track the improvement of cultivars regarding this particular species. Then for tuber yield, limited knowledge also exists on QTL mapping studies, according to that author. They have used conventional family-based linkage mapping to really detect the QTLs for YMV. So the GWAS strategy is also very important, to really understand the naturally occurring variants, and often robust and efficient markers are very important, even in identify significant loci and underlying causal agents involved, in the genetic control of these traits. So also, GWAS mapping is important, especially for the yield and YAM mosaic virus tolerance for YAM. So association of genotypic information with phenotypic traits of interest are useful, for selection of individuals with higher genetic value through marker-assisted selection. So the objective of this study was actually to dissect the genetic control of tuber yield and also the YAM mosaic tolerance in white YAM genotypes. So we used 406 clones that are at various stages of the breeding program. Some are parental materials. We have 49 that are at the advanced stage of the breeding, and then 321 that are early generations. These were obtained from polycross and nested designs, So we did phenotyping on these key traits, and then disease score was obtained, of course, following Forbes et al. 2014 protocol for the area under disease progress curve. It was done over time, monthly up to 6 months after planting, and then we use that protocol to work out the area under disease progress curve. Then for there are various analyses that we are for the phenotypic analysis. We can see all of them highlighted. I can't read through all of them. So broad sense irritability was also obtained using Robinson et al. 1949 rather. So the block values of genotypes for traits extracted from the best fit model were used as input for the GWAS model. So we can see also that leaf samples were collected from which DNAs were extracted from them using standardized protocol, and then the concentration quality of them were obtained using gel, agarose, and nanodrop. And we can see stepwise here how really the things we are done. And then we had to ship or send those samples to Canberra, Australia for that sequencing. So of the 20,000, things or have modified that we are obtained, about so I think 5,581 useful ones we actually used, those with useful SMP information. So for the population genetic analysis, we can also see detailed protocols that we have followed, standardized protocols that we have followed So I can't read, go through all of them, but we can see them through the slides. So the most likely value of the K was determined using the optimal subpopulation model as follows. They are also highlighted here. So basically, seven subpopulations were obtained based on that delta K value. And that one was consistent using the other techniques that we are used, because basically we use a structure harvester to have the structures obtained from them. Of course, the phylogeny tree also had to categorize those various clones into 7. And then even the PCA analysis also did likewise. So for the genome-wide association analysis, basically 6 multi-locus models we are used, and they are named here. And then for the percentage variations explained by the associated marker, also we can also see that those ones were obtained. Okay, so for the identification of mutative genes, haplotype estimation and marker effect prediction, we also used this window of 1 megabyte at 500 kilobase downstream and upstream, and then the linkage disequilibrium, to really assess, to make an assessment between the significant SNPs. We are also done using Shin et al. 2006. And then we also had to use this European Molecular Biology Laboratory, EMBL, EBI, also to try to pinpoint the genes actually to identify SNPs associated with those protective genes. So all pairwise linkage disequilibrium estimation between loci within a window of this, of each of these, also we are done in intercell 5.0. So these are also the other things that we are done. The linkage disequilibrium plot, the haplotype associated with significant QTL and others using standardized protocol. So for the result, we can see wide variation, for instance, for the area under disease progress curve. If you look at the range of values, the minimum and the maximum, you can see there's wide range variation. And then, of course, that one is depicted by the standard deviation there. We can see the broad sense heritability for both of the traits is really high because it's over 60%, over 0.6, so it's really high. And now when we look at the marker distribution across the chromosome, and we can see some values here and depicting the variability, the wide variability, even in the traits studied. We can see for the observed heterozygosity, for instance, the maximum over 0.6, and then the minimum is really 0.062 above. And then also you can see the mean value. You can see for the expected heterozygosity also exhibiting wide variability. And then for even the minor allelic frequency, and then even for the polymorphic information content, which are just all constituted genetic parameter estimates in the population studied. So for the various clustering techniques to really dissect the population structure, we can see that the delta K value was obtained at 1306.47, really high. The one showing the high peak here, justifying why, the clones were actually classified or categorized into 7. We can see similar pattern being shown here by the phylogeny tree, the 7 string pattern of the various genotypes, and we can also see how that one was obtained using the structure harvester, having also the 7 various groups of the clone study. So for the principal component analysis also follows similar pattern of the various clones that we are studied, that we are obtained. So now, for the genome-wise association, analysis of the two key traits or economic traits that we have studied, we can see that for youth, for instance, 9 chromosomes were actually identified with markers that were really significant in detecting putative genes. So we can see the markers here named here 4, 5 through to chromosomes. I mean, the chromosomes identified from 4 through 19. And then also we can see the same pattern in terms of the expected and the observed log values. So essentially, we can see that traits for this particular thing, we can see that there's like no inflation in the P values. You know, that indicates that structural relationship is adequate for GWAS analysis in this study, according to Yan et al. 2014. So also similarly, when we look at the Yam mosaic virus, you know, we can also see that about 5 chromosomes were identified, you know, that are actually depicting things for the Yam mosaic virus, you know, traits or alleles that are really depicting that one. So using those various multi-locus models, of the 6 multi-locus models that were used, 4 were very useful in identifying SNP markers, you know, significant SNP markers for the Yam mosaic virus and that of the tuber yield plant. So we can see here, oh my goodness, sorry. I think, okay, we can see here that, for instance, for the Yam mosaic virus, there are three markers that had negative quantitative trait nucleotide effects. You know, we can see them and their values are here for the percent contribution of those particular markers. They are also highlighted here in black. And also, we can see that the range of value for the minor allelic frequency or minor allele frequency range from 0.16 to 0.49. So it's really wide. Also, when we look at the tuber yield per plant, for instance, most of the markers exhibited significant, negative quantitative trait nucleotide effect, except, for instance, for the one on this marker 0.5 here that had this 0.03, there we can see its contribution is really high, about 8.47%, you know, contribution. So there are a few of those. Then when we look at the one by pillar MEB, is 0.02, then of course its contribution is 0.39, you know. So otherwise, most of the markers actually identified in this particular thing exhibited negative quantitative trait nucleotide effect in this study. Oh my goodness. Okay. Now here, okay. Here I'm basically showing the linkage disequilibrium of the pairwise linkage disequilibrium between the SNP markers located on the different chromosomes identified for the tuber yield. We can see that essentially for those 9 chromosomes, the heat map structure here is actually showing the high display of genetic correlation ranging from 0.3 to 0.85 with specific SNPs in the vicinity of the peak adjacent to the putative genes. So actually putative genes were identified whose functions have been reported in some crop species, you know, and that will actually help, you know, in further analysis or studies of the genetic architecture of even yam. Now we look at the Yam mosaic virus severity, the area under disease progress curve. Also, about 5 chromosomes were identified here, you know, genomic regions that are associated with this particular trait and also with high association. So These ones are highlighted here. So with a haplotype analysis, we found out that for the tuber yield per plant, there are about 12 markers that actually yielded about that has significant associations when you look at values here, probability adjusted significant values. There are about 12 markers for yield, and then for that of the yam mosaic virus, of the 5, 2 actually exhibit a significant association, based on the p-values for that particular trait. Also, for the box plots display, the box plot display of the effect of the significant markers, for the yield, we only show like 6 significant ones here, those that really showed variance. Like for instance, variance CC and CT were identified to be associated with genotypes with higher tuber yield. In contrast to that, TT and AT were found to be associated with lower tuber yield in this particular study. Then for the yam mosaic virus, also we identified the AA variants A and AG to be linked to lower predicted YMD values. Then on chromosome 16, we identify variants AA and AT, to be linked to lower predicted YMD value. So for the population structure analysis, I already highlighted that 5581 SNPs differentiated these 7 distinct sub things. Then for this marker trait association analysis using multi-loci, 17 QTNs we identify for the tuber yield per plant, and then 5 for the yam mosaic, with various effects. Then of these 17 that we have detected, they were really detected on 9 chromosomes. Then those ones were identified on 5 chromosomes. Then the variants responsible for predicting higher yield and low virus severity scores and the breeding panel, they are detected through marker effect prediction. Then gene annotation for the significant SNP loci identified several essential putative genes associated with growth and development. Putative genes like expansin, expansin was detected even in the tuber. We had the OX and the IAA gene also identified their modulating growth, for the yam mosaic virus. We identified Gemini virus, AL1, and a number of others were actually identified, to modulate or control even the expression of yam mosaic virus disease. Now, so all those ones support the view that the multi-loci models are really useful in identifying QTNs and candidate genes in plants. That one had been previously suggested or pointed out by Kari et al. 2020. So we can see that in this study, useful genetic variability exists in the 406 genotypes studied. And also that the genetic architecture of tuber yield plant and that of the yam mosaic virus are regulated by various QTNs, although they are unevenly distributed on the 20 chromosomes of the YTN. Also, among these 4 the multi-locus models, that we are used, the PK, WM, EB, and PLA and EB are most robust in identifying more QTNs. And then also they are situated as SNP markers could be potentially employed for targeted and associated tuber yield plant and yam mosaic virus resistance in YTN. Now, so the information actually from our study could help design new breeding strategies to forward superior alleles for tuber Yoruba plant and yam mosaic virus in future marker-based breeding. Also, the chromosomal regions identified controlling these study traits could be exploited for selection and effective pyramiding of favorable alleles and white yam population improvement. Also, we can recommend that findings are relevant for population improvement of desirable tuber Yoruba plant and yam mosaic virus traits using marker-assisted breeding and haplotype-based schemes. So I want to thank University of Ghana, Ahmed. Really appreciate that's the WACI program, West Africa Crop Improvement Program, for their great input. The IITA, been so great in raising scientists, helping students. I have benefited so much from them, and that's really so great. For the Africa Yam project sponsored by the Bill and Melinda Gates Foundation. Sponsoring this work, we are so grateful. The government of Sierra Leone, Sierra Leone Agricultural Research Institute. I am so grateful to the IDRC who also partly funded this program. And then to STEMU for organizing, putting this program together and giving us this opportunity to make the presentation. The photo here you see is just a cross-section of the research team at IITA. I am here actually. I want to thank you to you, my valued audience, for your keen attention. Thank you very much.